What has been the struggle or the search for the sacred graal, I call it, stem cell in cardiovascular disease over the last 10 years. Can I advance or will you advance? Yeah. I'm an interventional cardiologist, so I'm the guy that goes to the emergency room and the cath lab to open up vessel when somebody has a heart attack and place a couple of stents, or when the patient has not been so lucky to get soon enough to the cath lab and has already had time to develop permanent damage, then I put a defibrillator or I put a pacemaker trying to get the heart going again. What we have learned in cardiology is that when you have interruption of blood flow that is causing a myocardial infarction, the best thing to do is to open up that vessel as soon as you can. We have now a absolute order to have a door-to-balloon time, as we call it, less than 90 minutes across the United States and also in Europe. And because we know that if we can open that vessel within that time, that is what will repristinate the function of the heart. And so there is not much time to inject stem cells in that setting. You understand, what you want to do is to get there, have a crew that is on call 24 hours a day, and get your patient immediately to the cath lab and then open the vessel. So in this type of setup, it's really not feasible to inject stem cells. And the initial experiments that were done in the first 4 or 5 years trying to inject stem cells at the later time with a balloon and with stent, as one of the previous speakers was mentioning, has not really yielded much of an advantage, nor in terms of function, nor in terms of decreasing the stenosis stent, as it was said. At least the results have been so mixed that for 50 studies that have shown a benefit, there were 50 studies that showed no benefit. So this has been the struggle that we have been facing. And now, therefore, we have moved our research at the stage of heart that have already developed what we call a cardiomyopathy, or if you will, a negative, unfavorable development, where because of the development of a scar tissue, you already, because of the Laplace law, the pressure inside the cavity that is getting bigger is causing a progressive enlargement of the heart due to this substitution of a scar in place of an originally healthy muscle. Next. So, as I was mentioning initially, we were injecting stem cells in the coronaries, and we tested this as a possible approach. Another approach was instead to use an electromechanical map. This is a NOGA system. Essentially, you have the patient that is placed between two large magnets, so that when you have a catheter that has a metal tip, you know exactly where the catheter is inside the left ventricular cavity, and you know if the catheter tip is touching the left ventricle, and so you know if the part where the catheter is mechanically functional. But also you can record the electrical voltage at that specific site. So you can create truly a map of the heart where, for instance, the red is bad, there is no motion at all, but you can tell if there is still electrical signal indicating that there is some potentially viable muscle that could be salvaged or brought back to a better function. And so then you can operate an intramyocardial injection of the stem cell. Next. We were the first institution, thank you to this friend of mine, one of our interventional cardiologists and is the director of our stem cell center, Emerson Perrin, who's from Brazil. And so in 2003, Emerson, through his former professor, initiated this research because in Brazil it was much easier due to the obvious lack of an FDA to get this sort of research going. Next. And now we are part of a consortium of 7 centers in the United States. We have been testing, we have been participating to several trials. It's Stanford, Mayo Clinic, Indianapolis, Louisville, Gainesville, and us. And that's a way that we need to proceed because, as you heard from previous speaker, it's very difficult to go forward through anecdotal experience. We need to know what works, what doesn't work. Go on. So this is an example again of you see here the voltage map. You see here instead the mechanical map. Nothing moves, but there is some sign of potential salvageable muscle. So you inject the stem cell, and then eventually you see if some of the red has gone away, and it has gone away. Some of the good muscle has been able to come back. Next. Unfortunately, it has been very difficult to prove clinical improvement in some of these trials. You see here that what you want to look is the functional capacity of an individual. You want to see if his maximal oxygen consumption that is proportional to his ability to walk has improved in the treatment compared to the control group. And unfortunately, we have had an extreme hard time improving that in some of these trials.